Hello and welcome to another lovely edition of the program Teaching and Learning on Radio. Teaching and Learning on Radio is a program that comes your way every weekday on Prestige FM and some other radio stations. And it's brought to you by the Ministry of Education right here in Niger State. We have lots of subjects that we treat every week, every day of the week. We always try to see how we can give the students at home another opportunity to learn while they are home. You know, the pandemic is everywhere. The coronavirus pandemic is everywhere. And so the shutdown and the closure of schools and companies actually made the students are at home right now. Those preparing for WAEC, NECO, and other examinations, I think it's an opportunity for them to stay at home and learn while listening to radio. The teachers, the facilitators will be right here in the studio with us. They will tell us about a particular subject. They look at a subject and a topic, and the phone lines will be open for you to actually call in or probably send a message and ask your questions, and you will definitely learn something. So, well, it's for students at home, and it's brought to you by the Ministry of Education right here in Niger State. Teaching and Learning on Radio. Teaching and Learning on Radio it is. Well, today we have lots of things to learn, and we have our facilitator in the studio. You're welcome.
Hello, students at home. You are welcome to yet another segment of biology lesson. In our last lesson, I told you that SSCE biology is divided into 3 papers. The objective paper, we have the theory, and the practical paper. Today, we shall be looking at the practical aspect of biology, and I will treat some hints on biology practicals as well as some basics of biology drawings. Biology practical, unlike chemistry practical, where is different. In chemistry practical, chemical A is usually mixed with chemical chemical B to form a product, and in physics, measurements are taken using instruments. But in biology practicals, it is a different ball game altogether. In biology practicals, specimens are involved. Now, what is a specimen? A specimen is an example of a plant or an animal which represent the total plant type or the total plant and the animal respectively. Now, in biology practical, students are always advised or asked to answer all the questions, and also specimens are usually provided, displayed, and labeled accordingly using capital letters. The letter written against any specimen signifies that specimen. So students are always asked to study the specimen carefully, because they will use the specimen to answer certain questions. And also, students might be required to identify specimens. And if you are asked to identify specimen, what you do as a student is to either state the common name of that specimen or the botanical name. In stating the common name, for example, we have cockroach. Cockroach is an organism. It might be a specimen. If you want to identify cockroach, what you do is to write the common name, which is cockroach. Or you can write the botanical name, which is Periplaneta americana. But remember that if you are identifying specimen, it requires one word answer or botanical name, and therefore it must be spelled correctly for you to score. If you miss any letter or you replace a letter with another letter, you are not going to score. You are going to lose the mark. So students at home, learn how to spell one word answer or technical word correctly for you to score. Now, students might be asked to identify specimen with reasons. If you are asked to identify with reasons, what you are expected to do here is this, is to use the characteristic feature of the class that specimen belong. For example, if you want to use, or if you're identifying cockroach with reason, cockroach is an insect. So what you do, you are to use the characteristic feature of the class of cockroach as your reason. You state it out for them to understand. And for example, a cockroach is an insect, and it is a known fact that all insects have 6 legs. This is a unique characteristic feature for all members of that class. There is no organism in that phylum, Arthropoda, that possess more than 6 legs. All insects possess 6 legs. So it's a common feature for all members of that class. And also, they have jointed appendages. So you can use some of these points as your reason if you are asked to identify with reason. Now, if the specimen you are identifying is a plant part, maybe a fruit, a flower, a leaf or seed, all these are parts of plant. What you do is to indicate the part of that plant if you are identifying. For example, we have maybe tomato seed, we have tomato leaf, we have tomato fruit, and so on and so forth. So if you are identifying, you must state it. If you omit the part of that plant, you are not going to score any point. If, for example, you are identifying tomato fruit and you just write tomato without the fruit, even though the spelling of tomato is correct, you are not going to score anything for omitting that fruit, because tomato has different parts. Is it the seed you want to identify? Is it the fruit you want to identify? The examiner is not going to think for you. Please, students at home, take note. Now, if the specimen you are identifying is a leaf, the students might be required to answer the following questions. 1, they might ask you to classify the plant into its division and its class. They can equally ask you the type of leaf arrangement. They can ask you the leaf shape, the leaf margin, or the leaf venation. The leaf venation, this is the type of vein that the leaf is having. So let's take, for example, mango leaf. If we want to classify mango with this point, we can say that mango leaf belongs to the division Angiospermatophyta, and it belongs to the class Dicotyledoniae or Magniliopsida. So you have to classify, and the type of leaf arrangement in mango is alternate leaf arrangement. So if you go to a mango tree, the leaves are alternating each other, or you can say alternate arrangement or spiral arrangement. The type of leaf shape is known as lanceolate. You can call it lanceolate or sword-like. It is looking like sword. And the leaf margin is entire, or you can call it smooth, or it is wavy. Whichever one you use, you are correct. And the type of leaf venation, that is the type of vein the leaf is having, is net venation. Now, if what you are identifying is a plant flower, they might likely ask you the following questions. They might ask you to give the names of the floral parts. Flower has several parts, but the main part of a flower is known as the floral part. And most flowers, they have 4 floral parts. We have 1, the sepals, which is collectively known as the calyx. We have the petals, which is another one, collectively known as the corolla. It is not corona, it's corolla. And we have another floral part, which is known as the stamen. The stamen is the male part, and you can call it andresium, or the female part, which is carpel or pistil, and you can call it gynecium. So these are the 4 major floral parts of flowers. Now they might ask you also to state or name the numbers of each floral part. And the flower of Pride of Barbados, for example, Pride of Barbados is a type of flower. The sepals, which is one of the floral parts, is 5 in number. The petals is equally 5 in number. Then the stamen, which is the male part, we have 10 of them. And lastly, the female part, which is the pistil, is just 1. Now they might ask you to name the sex of the flower, because there are some flowers that are male, there are some flowers that are female, and there are some flowers that are mixed, they are hermaphrodite. That is, they have both the male and female sex cell. That is the gamete. They have both pollen grain and stigma. They might equally ask you to state the symmetry. That is the part when you cut the flower into two. For Pride of Barbados, for example, the symmetry is bilateral. It is bilaterally symmetrical. That is, you can cut it into two equal halves. Now you might be asked to give the agent of pollination. In this, they are talking about the transfer of pollen grain from the one plant part, male part, to the stigma, either of the same plant or of the same flower or of different flower. Now what they are asking you is the agent. That is, what is responsible for the transfer. What is that thing that will pick up a pollen grain from a flower to the stigma of another flower or of the same flower? For Pride of Barbados, for example, or most flowers, we have bees. Bees are very good in that. When they go to suck nectar, they will end up pollinating the flower. We equally have butterfly. Now, if the specimen, that is, if from the specimens that are provided, if you have two different types of flower, they might ask you to give the observable features, observable similarities, or observable differences of the flowers. Here, what you are expected to do is to observe the specimen. Viewers or students, I believe you are taking note. Observe the specimen. Then use the points noted to state out the similarities or the differences. In this case, you are not expected to use the general anatomy. No, that is what most students do. They usually use the general anatomy. No, you can only use what you observe, what you can see physically. That is what you are going to use to state the similarities as well as the differences between them. And also they might ask you to cut the flower into two, maybe the longitudinal section, that is cutting it vertically to reveal the internal structure. If that is done, they must ask you to draw the longitudinal section of maybe any flower, the one that is provided. So the students are at home, learn how to draw and label very well. Now, if the specimen that is provided is a fruit, for example, they might ask you to give the placentation. What is this placentation? It is just talking about the way the seeds are arranged in the fruit. Because fruits, there are seeds in them. And the arrangement of the seeds in fruit differ from flower and from fruit to fruit. And this arrangement depends on the arrangement of the ovules inside the ovary. Flower, we all know, develop into fruit. Then the ovules of a flower develop into what? The seeds. So for maybe orange fruit, the seed and the placentation is we have axile placentation in orange, axile placentation, and they might ask you to give the type of fruit. Orange is also known as berry. They might ask you to give the disperser, agent of disperser. What is that organism or that thing that is responsible for carrying the fruit from one place to another to make it spread all over the place? Man is one good example. Man can just suck the juice from the orange and throw away the seeds. The seeds with time will germinate and develop into a full orange. Now this brings us to the end of plant biology. Let us take a look at the animal biology. The kingdom Animalia, as it is called, is divided into 9 phyla. 2 of these phyla is common in SSCE. We have phylum Arthropoda. This phylum Arthropoda, they have segmented bodies and maybe jointed appendages, 2 or 3 body divisions. And this phylum has 4 classes. We have class Insecta, which is the class of insects. All insects belong to this class. For example, ant. Ant is an insect. We have another phylum, which is another class, class Arachnida. This is the class of spider and maybe scorpion. I know some of you at home, or the students at home, you might be wondering, why is it that spider is not under the class of insect? Spider is not an insect. Students at home, take note, because spider has 8 legs, 8 working legs, but all insects, they have 6 working legs, and that is why spider is not classified under class Insecta. Now the third class under phylum Arthropoda is class Crustacean. This is the class of crayfish and prawns. And the last class under phylum Arthropoda is class Myriapoda. Myriapoda. This is the class of the millipedes and the centipedes. Million legs and 100 legs, as people used to call them. Now, the second phylum that is common in SSCE exam is phylum Chordata. Phylum Chordata. This phylum, all organisms or all animals that have backbone, vertebrates, they belong to this phylum. There are some animals out there that have bones in them. We call them endoskeleton. And this phylum is divided into 5 classes. We have class Pisces. This class Pisces encompasses all types of fish. All fish belong to this class, e.g. tilapia fish. I am using tilapia because it is very, very important. If you are asked to identify tilapia fish in the examination and you just write fish, you are not going to score. You must identify it. You must tell us tilapia fish, because we have different types of fish. Now, the second class in this phylum is class Amphibian. The class Amphibia is the class of frogs and toads. They equally have backbones. Now we have another class, the third class, which is class Reptilia. This is a class of reptiles. All reptiles belong to this class. And example, we have snake, lizards, and tortoise. All of them belong to this class. Mind you, I didn't say class reptile, but class Reptilia. It is very, very important. Even the amphibia is class Amphibia without an N at the end. Please take note, students at home. It is very, very important. These are silly things that make students to fail practical biology, because they take note of spellings and so many things. Now, the fourth class under this phylum is class Aves. This is the class of all birds, be it birds that fly, birds in maybe dogs, poultry. All of them belong to this class. And the last class under this phylum is class Mammalia. I didn't say mammal, but what? Mammalia. And this class, there is one common or unique characteristic feature of this class. They possess mammary gland. That is, they feed their young one with milk. So any animal you know that feeds its young with milk belong to this class. For example, we have rats. You and I, students at home, you belong to this group, class Mammalia. So with this, we've come to the end of the animal part of biology. Now I want us to take a look on the basics on SSCE biology drawings. Why are we taking this? It is very, very important, because drawings are inevitable in biology practical. There is no how you can do without drawing. You must, is a must, to draw any specimen, maybe 1 or 2, 3 specimens, as the case may be, in any SSCE biology practical. So it is very, very important for you to take note of some of the basics. Now we have this. The point we have here is this. Drawings should be simple and neat with minimal error, and they should have neat labeling. When we talk about neat labeling in biology drawing, 2 things are involved. One, we have observing a ruled guideline. What are we talking about? We are talking about labeling using what? Ruler. You must use your ruler to draw the guideline. You don't have to use freehand. If you use freehand to draw the guideline, you will be penalized. So use your ruler. And the second point under neat labeling is horizontal labeling. If you are labeling your diagram, you label flat. That is what we mean by horizontal labeling. You label flat so that it will be parallel to the ground. So another point is drawings should be complete. When we talk about complete diagram, we are talking about diagram that is well labeled, diagram that have a title or heading. Students are fond of drawing without giving it a title. It is very, very important for you to give a title. If, for example, they ask you to draw the dorsal view of a fish, what you are expected to do, your title should be like this: diagram of the dorsal view of a fish. You must state it, and it should be at the top of your drawing. Another thing that you must observe under complete drawing is the magnification. The magnification should be written by the side of your diagram, maybe times half, times 1, times 2, as the case may be. And when you talk about magnification, you are talking about the size of your drawing, which is the image, and you compare it with the size of the specimen provided, which is the object. If your drawing is 2 times bigger than the specimen that is provided, then your magnification will be times 2. You must indicate it. It is very, very important. Now, all diagrams should have a size. All SSC exams, they ask students to draw, and the length of the diagram is usually given. They may ask you to draw 8 to 10 centimeters of any specimen. What you are expected to do is to get a ruler, go to the page where you want to draw, measure. You must measure. Students think examiners, they don't measure. They measure. You have to measure the space and make sure it is not less than 8 centimeters, and it should not be more than 10 centimeters. It should be between. Now, all diagrams, the guidelines should not cross each other. What we are saying is this: when you are labeling, you should not allow your guideline to cross each other. You are going to be penalized. If you label maybe eye in a fish and you want to label the operculum, the guideline for eye and operculum, they should not cross each other. If not, so you are going to be penalized. And diagram should be very thin. You use sharp pencil to draw so that it should be thin, and it should be straight. It should not be broken, that is woolly, and it should be continuous. And the diagram must not touch, the guidelines must touch the structure you are labeling. What we are saying is this: your guideline must touch what you are labeling. It should not be left afloat on air. No, let it touch it. If you fail to allow it touch it, then you will not be given the mark. And because of lack of time, with this we've come to the end of today's lesson. So students at home, if you have any question to ask, please, you can join us on our WhatsApp group with this number 09029282040. Again, 09029282040. And please, I would like to give you an assignment. Now, I want you to classify the following specimen into kingdom, phylum, and class. 1, I have Ixora leaf. Classify it. Another specimen I have there is water lily plant. Then you should equally classify grasshopper, and lastly, you classify fish. Draw the dorsal view, 8 to 10 centimeters of a tilapia fish, and state the functions of each labeled part. Thanks for listening.
That was a wonderful presentation by our facilitator. He taught us a whole lot, and I learned as well, so I believe you've learned something this lovely edition of the program, Teaching and Learning on Radio. Well, join us again next time, that's tomorrow. Join us again and for another lovely edition of the program. You never can tell the subjects coming up, so just be on the lookout. And children out there, and not just children, parents, I urge you to encourage your children to listen to the program. As they stay at home, they learn as well. All right, thank you so much for watching and listening. Bye-bye.